So the problem we're looking at today is why exercise-mediated increases in PGC1 alpha are impaired in obese individuals compared to lean. As you can see on the graph on the right, there is more PGC1 alpha protein in the lean individuals compared to the obese. And if you look at the graph on the left, it shows that there is more PGC1 alpha mRNA in lean individuals compared to obese when exercising. So our mass that we're going to look at is the PGC1 alpha mRNA. The outflow of this is translation into the protein PGC1 alpha. The inflow to PGC1 alpha mRNA is transcription via the transcription factors in DNA that's activated by the active PGC1 alpha protein. This process is also a cyclical pathway where the PGC1 alpha is activated through CMK, P38, AMPK, and SIRT1. So if we look back at our problem, we can see that there is more PGC1 alpha protein in the lean individuals compared to the obese. This means that the outflow had to increase the amount of protein there was. However, if we also look, we see that there is more PGC1 alpha mRNA, which is our mass of interest, and that also increased. So if the mass increased and the outflow increased, this must mean that the inflow to the mass increased more than the outflow did. So we want to know is how the inflow is increasing more. We first wanted to look at the activators to PGC1 alpha.
So first we're going to look at the PGC1 alpha activators to see if that will elicit a change in PGC1 alpha between the lean and obese individuals. However, because they were exercising at the same relative intensity, the ADP, AMP concentrations should be the same between each group, the epinephrine release during exercise should be the same between each group, and also the calcium secretion during exercise should be the same each group. Those three things both activate this PGC1 alpha pathway via CAMP kinase, P38, SIRT1, and AMPK. Therefore, these PGC1 alpha activators are actually not the case. Furthermore, you could argue that lean individuals have an improved energetic status of muscle cells. Therefore, you'd expect obese individuals to have higher pools of AMP, which would elicit a stronger activation of PGC1 alpha and therefore PGC1 alpha protein. And this is actually the opposite. So it's very much likely that the PGC1 alpha activators are not the case. Therefore, our hypothesis is that the larger exercise-mediated increases in PGC1 alpha mRNA, and therefore PGC1 alpha, seen in lean versus obese individuals, is due to increased expression of PGC1 alpha's transcription factors. A paper completed by Hanschen and friends found that myosin enhancing factor 2, or MEF2, is a PGC1 alpha transcription factor. The two graphs on the left indicate this being a transcription factor as it activates the PGC1 alpha promoter, and they actually illustrated this in a nice schematic over here showing you the proof that MEF2 is actually a PGC1 alpha transcription factor. Therefore, if we were to include MEF2 into our flow chart, we would see that MEF2 is over here representing an inflow along with DNA and active PGC1 alpha protein to create PGC1 alpha mRNA. Therefore, our revisit hypothesis states that the larger exercise-mediated increases in PGC1 alpha mRNA, and therefore PGC1 alpha protein, is due to increased expression of MEF2 in the lean individuals. In other words, the smaller exercise-mediated increases in PGC1 alpha mRNA, and therefore PGC1 alpha protein, is due to decreased expression of MEF2 in obese individuals. We then looked into the literature to see if there's any evidence on differences in MEF2 expression between lean and obese subjects. So a paper completed by Krunkhorn et al. Had two conditions with mice, where one condition was fed a high-fat diet and one condition was fed a regular low-fat diet. This high-fat diet represents obesity, as high-fat diet can lead to obesity, or as the paper mentions, acquired obesity. The results in this paper, I'm going to first draw your attention to this paragraph on the left here, it states that the expression of MEF2C and MEF2A were significantly decreased by palmitate, which is that high-fat diet. In addition, MEF2 DNA binding, the binding site was also decreased by palmitate. There was no actual figure in this paper, so we illustrated our own just to kind of give you a nice illustration of this. On the left here we have the control group, and on the right we have the high-fat the high-fat diet. And as you can tell, the blue bar representing MEF2C expression, orange bar MEF2A expression, and gray being the MEF2 DNA binding site, those were all significantly decreased in the high-fat diet, which again represents obesity. Therefore, if we were to include this into our flow, we would show that palmitate, or again obesity, decreases the expression of MEF2 and its associated binding site. So therefore, we could state that obesity impairs the inflow by decreasing the MEF2 expression, and therefore can decrease the PGC1 alpha mRNA pools in obese individuals versus lean. So if we were to conclude this, bringing back to our original problem, we could say that palmitate, which again indicates obesity, significantly decreased the expression of MEF2, A and C, and their associated binding site. Bringing this back to our original problem, we could state that therefore, the obese individuals may have experienced lower exercise-mediated increases in PGC1 alpha mRNA, and therefore PGC1 alpha protein, because of decreased expression of MEF2, which is related to obesity.